Good morning and welcome to all of you. B.Sc. second year, semester third, subject Fishery Science, paper 6th, Fish Disease Management. From this paper, we have studied few articles, few fish diseases, few fishes or few pathogens. These are to be attacked on the fishes and they cause the harmful effect in fishes, and it is to be again harmful for the fish culturists or the fish farmers. The next important article which is to be studying today, and that important point, that is the worm diseases or the Helminth diseases. Earlier we have seen elaborately the important pathogen fungi is fungal diseases. Important pathogen bacteria, that is number of bacterial diseases. Then important pathogen protozoan, that is protozoan diseases. All these diseases, these are to be seen. Then we have studied worm diseases or the Helminth diseases. Worm diseases or the Helminth diseases. Those worm Helminth, these are to be found on Earth planet. These Helminths, these worms, these are also act as pathogens. These are also attack on the animals and they cause the different adverse effect on living animals as on the, that is the fishes or the fish cultures. Generally the four main groups of worms on fishes. Generally four main groups of worms. These four important main groups of worms, these are to be attack on fishes and cause the pathogens, cause the fish diseases. Which are these four main groups? The first, flatworms. First group is the flatworms. This flatworm, these are also called as trematodes. Flatworm is also called as trematodes. Second group of worms, this is called as a tapeworm. Tapeworm. This tapeworm, it is also called as, it is also known as, that is the cystodes. Tapeworm, cystodes. Third group of worms, it is called as roundworms. Roundworms, these are also called as nematodes. Roundworms are the nematodes. And the last or the fourth group of worms, it is called as thorny-headed worms. Means trematodes, cystodes, nematodes and thorny-headed worms. These four group of worms, these are to be commonly attacking, these are to be generally attacking on the animal species, on the animal fishes in aquaculture as well as in the capture fishes and culture fishes. The worms generally affect the internal organs of the fish, particularly the intestine. Where these worms, these are to be attacking like that of the other fungal pathogens, bacterial pathogens, protozoan pathogens. These worm pathogens, these are to be not attacked. These worms, these are to be not affected on external part of the body of the fish, but these are to be attacked, these are to be affect, these are to be attacking on the internal part of the body. And that's why these worms, these are to be more serious and more dangerous, because those pathogens which is to be attacking on external side, ectoparasite, these are to be, we have removed easily, we can seen easily. with naked eyes. But those are the endoparasite. Endoparasite, the parasite, these are to be attacked, these are to be affect on the internal part of the body, such as intestine, such as alimentary canal, then it is to be called as the endoparasite. Worms generally these are to be attack, worm generally these are to be affect on the internal part. Internal part, which are the internal part? The most attacking internal part is the alimentary canal. Most attacking part is the intestine, alimentary canal, just behind from the mouth to all alimentary canal, these are to be severely affected, seriously affected by these worms. Hence this intestine or internal part, these are to be more affected. Among worms, two types of parasites. Among worm, two types of pathogens, these are to be found, these are to be occur. Generally parasites are ectoparasite attacking on external side, parasite these are to be attacking on the internal parasite, endoparasite. Among worms, two types of parasitic worm these are to be found, which is to be occur. One is the monogenetic worms and second is the digenetic worms. Monogenetic worms and digenetic worms. Among these two worms, the monogenetic worms, these are to be more dangerous and these are to be more serious for culture, for the different animals or the different organisms. Why it is to be monogenetic more dangerous, more serious? Because name itself indicate that. Name itself, it is to be suggested its meaning. Monogenetic worms. Monogenetic worms, those worms, those flukes, worms, these are also called as flukes. Those worms or those flukes which complete their life cycle, those animals, those pathogens which complete their life cycle within a single host, within a one host or single host, and then they can transfer directly from one host to another host, from one animal to another host, and these are to be cause the disease, these are to be cause the pathogen. That worms, these are called as monogenetic worms. Monogenetic worms. That monogenetic worm, that may be monogenetic trematodes, that may be monogenetic nematodes or monogenetic cystodes. These are more serious, these are more dangerous because they complete their life cycle within single host, within single host. Hence they can rapidly grow, they can rapidly reproduce, and they can easily attacking to the other animals from one host to other host. These are to be direct transfer. Hence this monogenetic trematode, these are to be more dangerous, more serious for the aquaculturists, for the fish culturists. Then which are the examples of these monogenetic trematodes? Which are the examples of these monogenetic worms? The first example, that is Gyrodactyalus, and second is the Dactylogyrus. These two parasite, these two pathogens, these are the monogenetic worms. They complete their life cycle in single host or the one host. Second, digenetic worms. Digenetic worms. Di means two, genetic means, gender means two species. That is, those flukes or those worms, those flukes, those worms who complete their life cycle in a two host, those pathogens, those parasite which complete their life cycle. For completion of their life cycle, two hosts these are to be essential, two hosts these are to be required, two hosts these are to be essential, two hosts these are to be required. If host other than the fish, if the host other than the fish, secondary host, primary host, that is one animal. From this animal they can reproduce, they can generally a breeding, these are to be done. And secondary host, if the secondary host other than fish, primary host is the fish and secondary host is to be not fish but other animals. Secondary host, their life cycle never gets complete. Suppose among these two hosts, any one host these are to be die, any one host these are to be complete lost, any one host, these are to be complete eradicated, control this disease, these are to be controlled. But suppose the worm is to be a monogenetic worm, for completion of their life cycle only one worm, only one parasite, these are to be essential, it is to be called as monogenetic. And digenetic, those which is to be required two hosts, one is the fish, second host is the other animal, that may be cow, that may be buffalo, that may be sheep, that may be goat, that may be man, or that may be any animal. To complete their life cycle, these two animals, suppose secondary hosts, these are to be lost, their progeny, their reproduction, these are to be stopped, and spreading of this disease, these are to be completely stopped, these are to be completely avoided. That's why these digenetic worms, these are to be no serious, no dangerous as compared to, or it is to be less serious, less dangerous as compared to the monogenetic worms. That is, their life cycle never gets complete, and their control is to be easy, their regulation, their maintenance, these are to be easy, and we can control such pathogens, such parasites. That's why digenetic trematodes or the digenetic worms, these are to be more easier and more become to control the pathogens, control parasite, we can easily control, we can easily become controlling, that is the digenetic worms. Digenetic worms, these are the two types of worms, these are to be found in the fish species. Then the worms include the following diseases such as trematodes. Which are the trematodes? These trematodes, these are to be important, these trematodes, these are to be most useful. The first monogenetic worms, that monogenetic worm is trematode is the Gyrodactyalus. Gyrodactyalus disease. This is the most important and most dangerous disease. These are to be occur, these are to be found in fishes. Gyrodactyalus disease. Then we have seen the disease Gyrodactyalus. Gyrodactyalus. This disease is caused by monogenetic trematodes, Gyrodactylus elignans or the Gyrodactylus salaris. This disease is caused by monogenetic trematodes called as Gyrodactylus elignans or the Gyrodactylus salaris. It is one of the most common diseases in nursery and rearing ponds in Indian waters. This is the one of the most important common diseases in nursery and rearing ponds in Indian waters. In Indian waters, especially in nursery ponds and rearing ponds, this disease, these are to be occur. This disease, these are to be found. That is the Gyrodactylus elignans disease. This disease starts appearing in ponds during monsoon season. When this disease, these are to be occur, these are to be found. This disease start appearing in ponds during monsoon season. Nearly 85 species of this fluke have been reported in fishes. How much species of flukes these are to be reported? This disease, these are to be occur, these are to be appear in monsoon season, in rainy season, and near about 85 species, means near about 85 various types of these flukes have been reported, have been attack on fishes. However, the most widely distributed species is Gyrodactylus elignans. The most important and widely spread fish pathogens, fish trematodes, that is the Gyrodactylus elignans, which is to be found, which is to be occur. See the figure. In this figure, we can observe, we can see the internal part of the fish, visceral organs of the fish. This is the heart, and behind the heart, this is the visceral organ. These are the intestine. These are the, that is esophagus, stomach, intestinal bulb, and this is below to this air bladder, which is to be occur, which is to be found, which is to be seen a whitish color. Worm parasite infected on internal part. Because this disease, these are to be more serious. This disease, these are to be more dangerous. Why? Because these are to be affected on internal part of the body of fish. Internal part, which is the internal part? Just from the mouth, that is specially alimentary canal. Alimentary canal, these are to be comprises mouth. Mouth leads into esophagus. Esophagus leads into, that is buccal cavity. Pharynx. Pharynx leads into stomach. Stomach leads into intestine. Intestine leads into rectum, and rectum leads into anus. All these parts, these are to be found, that is in alimentary canal, these are to be attacked. And when these alimentary canal, these are to be lost, feeding, these are to be lost. Internal muscle, these are to be change their color. Muscle, these are to be feeding by this pathogen. Ultimately, muscle, these are to be weak in condition. Alimentary canal, these are to be weak in condition. Feeding, these are to be stopped. Ultimately, fish becomes die, fish becomes dead. That's why these are to be attacking on the internal part of the body of fish. See, fish infected by various parasites. At a same time, in these fish, number of other pathogens, number of other parasites, these are to be affected, which is to be seen here. Which are these number of parasites or pathogens? First is the anchor worm. These anchor worm, these are to be attacked just behind to the operculum on the skin. This is the lice, head lice. These are to be attacked on the just below to the dorsal fin on lateral side. And this is the leech. This is the leech. Means worms, anchor worms, lice and leech. These three pathogens, these three parasites, these are to be attacking on this fish. Hence, it is to be multi-parasite affecting multi-parasite. These are to be attacked. These are to be affected on these fishes. This is C. Fish shows the Gyrodactyalus parasite. This is the Gyrodactyalus parasite. This is the Gyrodactyalus parasite. This is the Gyrodactyalus parasite. Means on the internal part of the body, internal part of the fish or flesh of the fish, these are the parasites, that is the internal part. These are the internal pathogens. These are to be attacking on the internal part of the fish body, internal part of the animal's body. These are to be observed. These are to be seen in this figure. That is internal part. These are to be affected. These are to be seen. Then what is the structure of Gyrodactyalus elignans? How we can identify? How we can observe this Gyrodactyalus elignans? These are to be seen. These are to be understood. We can observe. Gyrodactyalus are small worms about 0.5 to 1 mm in length. What is the size of Gyrodactyalus? It is to be about 0.5 to 1 mm in length. Found on the skin, found on the gills and fins. Where it is to be found? It is to be found in skin, gills and fins. In these part, this pathogen, this parasite, these are to be found. Each species is host specific. Each species of parasite, these are to be host specific. Host specific, a specific host they can select, they can choose, and may even be specific to a particular site to the host. Not only a particular host, but a particular site. Some parasite, these are to be attacked only on caudal fin. Some parasite, these are to be attacked only on pectoral fin. Some parasite, these are to be attacked only on intestine. Some parasite, these are to be attacked only on gill side. Some parasite, these are to be attacked on stomach or the intestine. Means these parasite, these are to be species specific, as like it is the also a host specific, as like a particular site, also particular place. This parasite, these are to be found. These are to be okay. Under low magnification, in a light microscope, they can be seen as small transparent organisms showing twisting and squaring movement. This animal, these are to be shown under the microscope as seen as a small transparent organism showing twisting and squaring movements. The body is elongated and flattened dorsoventrally. Body is to be elongated and flattened dorsoventrally with the posterior and modified to form attachment organ known as hapter. The body is elongated and flattened dorsoventrally with the posterior and modified to form an attachment organ that attachment organ to the body of host, that attachment organ is called as the haptor. It is called as the haptor, and these haptor, these are anchor, these are to be attached to the fish flesh or the fish muscle. The interior end is narrower, and a number of lobes bearing mouth which open the exterior. Number of lobes bearing mouth which open the exterior. The mouth opening leads into the pharynx. The worms are hermaphrodite with both male and female sexual organs opening to the exterior through the genital pore. Posterior to mouth, eyes are absent. What is the character of this parasite? The mouth opening leads into the pharynx. The pharynx are hermaphrodite. Pharynx are hermaphrodite with both male and female sexual organs being to be exterior. Both male and female have a sexual organ separately, differently, and these sexual organs, these are to be opening to the exterior through the genital pore. Posterior to the mouth, eyes are absent. Posterior to the mouth and eyes are absent in this parasite. In this, that is parasite. Which is the parasite? Gyrodactyalus elegans. See, this is the Gyrodactyalus parasite. Shows haptor. This is the tail portion. 1, 2, 3, 4 part. This is the nucleolus, and these are the haptor. 1, 2, 3. These are near about 16 pairs of haptor. These are to be found, and it is to be circulated or the semi-circulated haptor. These are to be observed, and with this haptor, this animal, this parasite, these are to be attacked to the fish body, and these haptor, these are to be piercing into the skin, and then these are to be attached to the fish body, and after attaching, they can move from one place to another place. Structure of Gyrodactylus elegans. The haptor in Gyrodactylus is disc shaped. Haptor is to be disc shaped. See in the figure, this is the disc shaped. Haptor is to be disc shaped haptor. Haptor is to be disc shaped with 16 small marginal hooks, and a pair of haptor forms ventral efficient attachment organ which is found near to eye post, and it is convenient way to distinguish between Gyrodactylus and it is to be important Gyrodactylus and ectoparasite fluke. It gives birth to young ones which are already well developed, which are already well formed condition. These are to be occur. These are to form. These newly born young Gyrodactylus leave the parent to occupy a place on the host of the parents to be transmitted by contact to a new host. How this new host, these are to be contact? These newly born young Gyrodactylus leave the parent to occupy a place on the host of the parent to be present. Those hosts, these are to be attached to the parent. On this parent host body, this parasite, these are to be contact. This parasite, these are to be attacking, these are to be attached. This is the worm parasite infected on visceral organs. See, these are the haptor. These are the anchoring condition. These are the mouth-like structure which is to be observed, which is to be seen, and this is the tail portion which is to be bifurcated, and two lobes, these are to be seen, these are to be observed. On body, small pointed point, these are to be observed, these are to be seen on this figure, that is worm parasite which is to be observed. Infected fish gills show the Gyrodactylus parasite. This is the gill slits. Infected fish, and these infected fish, this is the Gyrodactylus elegans. These are the Gyrodactylus parasite. These are the Gyrodactylus parasite. These are the Gyrodactylus. These are also Gyrodactylus parasite. These are to be attacking. These are to be affecting on the internal parts such as the gills, and when gills, these are to be affected. Gills, these are to be fed. Gills, these are to be completely lost. Respiration, these are to be lost, and this animal become ultimately die, or these are to be dead. This is the again flukes of trots. This is the Gyrodactylus elegans. See this haptor. These are the disc-shaped haptor. These are the haptor. These are 16 haptor pointed. These are to be piercing into the skin of the host and they can attach, they can found. This is the young embryo which is to be found in this young embryo, which is to be observed, which is to be seen. This is the pharynx and this is the mouth. How the fish, these are to be taken, the food material with the help of mouth, pharynx, these are to be observed, and in the body, that is young embryo, these are to be found, these are to be observed. The figure is to be observed, figure is to be seen, that is it is to be found in trots, that is flukes of trot, these are to be observed, these are to be seen. Worm parasite Gyrodactylus salaris, not only the lignans these are to be occur, but Gyrodactylus salaris, this is also another worm, this is also another parasite, these are to be affecting, this is pharynx, then penis, uterus, two embryos, these are to be observed, one is to be there, second is to be there, one embryo, these are to be occur, then that is ovary, these are to be occur, testes, these are to be occur, and this is the opisthoheptor, these are the heptor, these are to, this is the colored animal. When we have seen exactly in figure, that is a broad figure, these are to be observed, but when it is to be, we have seen in a magnification in a microscope, then it is to look like such structure, that is the Gyrodactylus salaris, these are to be, means Gyrodactylus lignans and Gyrodactylus salaris, these are to be found, it is to be occur. Then which are the, just up to here we have seen the structure of the Gyrodactylus. Then symptoms, which are the symptoms of this Gyrodactylus, which are the symptoms, these are to be occur in the fishes. The first symptom, affected fishes show fading of normal coloration of skin due to the parasite, due to the effect of these worms. The first, coloration of the skin is to be lost, coloration of the skin is to be faint, and lastly fish become die. When faint color, these are to be observed, it is to be attack on parasite. Loss of scales, those areas, this pathogen, these are to be attached at that place, scale, these are to be lost, scale, these are to be fall down, and excessive mucus secretion. When scale, these are to be fall down, and excessive mucus secretion, these are to be observed, other pathogens, these are to be attack that part, and fish become died, these are to be infected. Second symptom, Gyrodactylus may live anywhere on the host. Gyrodactylus may live anywhere on the host, but it is usually most abundant on the fins, especially the dorsal and caudal, when the parasites are very abundant. When parasite, these are to be more, when parasite, these are to be maximum or more in concentration, at that time it is to be attack, it is to be affect on the which part, that is found on the dorsal and caudal fins, these are to be specially found. The fins become frayed and it is to be eventually worn out. Fish becomes frayed. Frayed means that is findage, these are to be brittle, these findage, these are to be easily cut down, these are to be decomposed down, and these are to be fall down. Eventually worn out, it is to be gadun padne, jala आपण म्हणतो किंवा निघून जाणे. Dorsal fin and pectoral fin, caudal fin, these are to be worn out. The dorsal fin, caudal fins are worn out. What is the effect? Because these fins, these are to be useful in locomotion and swimming. Locomotion, these are to be stopped. Swimming, these are to be stopped. Other enemy, these are to be attacked, and these are to be used as a food. That's why it is to be more dangerous symptoms. Next, infected fishes can often be seen tubing themselves against the dike or bottom of the pond to dislodge the parasite. Infected fishes can often be seen tubing themselves to rub the body to the bottom, to rub the body to the any material, any object. Why it is to be rub the body, any object, any material, to fall down the host, to fall down the parasite. These fish, these are to be identified. Infected fishes, these can be often tubing themselves against the dike or bottom of the pond to dislodge the parasite, to fall the parasite, to leave the parasite. These fishes, they can rub their body either bottom, either benthic, or the either different dikes areas, and this dike areas, these are to be seen, these are to be found. Next symptom, affected gill turns pale color. Next part, affected gills turn pale color. What is the important symptom? Pale color. When pale color, these are to be done, then incurrent and excurrent, these are to be not occur. Then in blood or in gills oxygen, these are to be not properly, in proper way these are to be taken, means respiration, these are to be occur. Respiration means that is a purification of blood, and gills, these are to be pale in color, these are to be not purified the blood, then ultimately fish becomes die, these are to be dead. Next character or next symptom, the affected skin gets covered by bluish slime. Those areas, those part which is to be affected by parasite worms, that part these are to be covered by bluish slime due to increased secretion of mucus. Due to the excessive mucus secretion, those areas, those part, this parasite, this pathogen, these are to be affected. That part, that area, these are to be covered by bluish slime, and this is to be due to the excessive mucus secretion. These are to be found, it is to be occur. Then to cure, to control this disease, what is the treatment or which are the treatment to minimize this disease for fish cultures, for prawn cultures or the carp cultures or the fish farmers? To minimize this disease, the treatment, these are to be essential, treatment, these are to be most important. What is the first treatment? The first treatment for the Gyrodactyalus disease. Dip the infected fishes in 1:2000 solution of acetic acid. First treatment क्या है? First symptoms treatment क्या है? Dip the infected fishes in 1:2000 solution of acetic acid. Those fishes, these are to be affected by Gyrodactyalus. Those fishes, these are to be affected by this Gyrodactyalus salines or lignans. These fishes, these are to be deep infected 1:2000 solution of acetic acid or sodium chloride, either acetic acid or sodium chloride, for about 5 minutes. It is to be recommended, it is to be important. For about 5 minutes, these infected fishes, these are to be deep treatment up to 5 minutes in 1:2000 solution of acetic acid or same proportion of sodium chloride. 5 minute fishes, these are to be deep bathing. Then this parasite, these are to be fall down, these are to be leave down. Second treatment, keep them in tanks containing 10 ppm acryflavin. Those fishes, these are to be infected by the Gyrodactyalus. Such fishes, these are to be collected and these are to be containing a tanks of 10 ppm acryflavin. Acryflavin, these are to be used, that level or the proportion of acryflavin is the 10 ppm part per million. This part per million acryflavin, these are to be used, or either 2 to 4 ppm of methyl blue dye. 2 to 4 ppm of methyl blue dye is to be used, it is to be utilized for 2 to 3 days. 2 to 3 days, these infected fishes, these are to be deep bathing, deep keeping up to 2 to 3 days of either in containing 10 ppm of acryflavin or it is to be containing 4 ppm of methyl blue dye. In this methyl blue dye or the acryflavin, these fishes, these are to be keep, that ultimately this parasite, these are to be fall down, these are to be removed. Another treatment, dipping the fishes in 1:5000 solution of formalin is recommended. Suppose first acetic acid is to be used, parasite, these are to be not controlled. Sodium chloride is to be used or salt solution is to be used, then also parasite, these are to be not controlled. Acryflavin is also used, it is also then not controlled. Methyl blue is to be not controlled, then we can use, that is the 1:5000 solution of formalin is recommended. 1:5000 solution of formalin, these are to be most recommended by the workers, by the scientists. And next treatment, these are to be done, that is treatment may be repeated after 2 to 3 days. Treatment, these are to be repeated after 2 to 3 days to ensure complete removal of parasite. To complete removal of parasite, these infected fishes, treatments may be repeated after 2 to 3 days to ensure complete removal of parasite. To complete remove the parasite, this method, these are to be adopted, this technique, these are to be adopted to minimize, to lower the effect of Gyrodactyalus ligni. These are the few treatment, these are the few treatment which we have seen. With this, we have completed first monogenetic trematodes. First monogenetic worm, that is Gyrodactyalus elegans. This monogenic this worm parasite, these are to be attacked in monsoon season in carps, major carps, exotic carps, especially carp culturists or the carp farmers. They threaten to this parasite, and these are the more dangerous because this is the monogenetic trematode. These are to be attacked on the fish fairly large, and internal part of the body, these are to be affected. Internal part, these are to be consumed, and we do not identify whether the animal is to be attacking or not, whether the parasite these are to be affected or not. That's why when we can see internal examination, then and then we can identify the parasite these are to be affected. Some symptoms and treatment we have seen here. Up to here, the first worm disease, that is Gyrodactyalus elegans, which is to be completed here. Thank you and welcome to again in next online class of fishery science. Again, thank you.